Welcome to the Department of Biology at the Paso Community College. Today we're going to be talking about PCR. What is PCR? You have probably heard about PCR right now that through the COVID-19 pandemic, everybody has been getting tested to confirm the presence of the SARS-CoV-2 virus. So pretty much right now we're going to be going and explain to you what is a PCR test and how is it done.
PCR is a very basic, standard molecular biology lab procedure that we do to identify the presence of a gene of any type of microorganism. Pretty much what we're looking for is the presence of a gene. It can be any microorganisms. It can be a virus, it can be a bacteria. In this case, it can be the SARS-CoV-2, right? So once we identify one of the genes that is very basic of that microorganism, we can confirm the presence of that microorganism in a person. So that's why it's considered a confirmatory type of test. And we're going to be also talking about the gel and some of the tips that I particularly use to make sure that I get a good quality samples and bands on my gel electrophoresis, that you can use for your presentations.
The idea of amplifying DNA came from Dr. Kary Mullis. Dr. Mullis was a biochemist, and while working with a method to detect mutations, he came up with a way to double his gene target. He noticed that this new method, he was able to double a short region of double-stranded DNA, giving him twice the number of DNA copies. His first successful amplification was an Escherichia coli plasmid, and received the Nobel Prize in Chemistry for this technique in the year 1993. To get started, it's very important to always take your time to disinfect your work area, and this is mostly to prevent any contamination of your samples. Before getting started with our PCR, we first need to grow and isolate our bacteria in blood agar plates. Our blood agar plates are gonna need to be incubated for a total of 24 hours. Once our plates are ready, we can proceed with the DNA extraction step, and you can actually follow any DNA extraction method of your choice. So what is a PCR? PCR stands for polymerase chain reaction. PCR is a method used to amplify a specific DNA sequence. This amplification of nucleic acids involves making millions of copies of a gene that can be detected in vitro over a short period of time. That is why sometimes this is referred to as a molecular photocopying. The real ability of the PCR is that it has the ability to amplify specific genetic targets. The PCR is a process that generally lasts around 2 hours. So how does the PCR process works? There are 3 main steps. Step 1 is the denaturation process. As in DNA replication, the 2 strands of the DNA double helix needs to be separated. The separation happens by raising the temperature of the mixture, causing the hydrogen bonds between the complementary DNA strands to break. This process is called denaturation. Second step is the process of annealing. Primers bind to the target DNA sequences and initiate polymerization. This can occur only once the temperature of the solution has been lowered. 1 primer binds to each strand. Number 3 step is the extension. New strands of DNA are made using the original strands as templates. As DNA polymerase enzyme joins free DNA nucleotides together, this enzyme is often Taq polymerase. This enzyme originally is isolated from a thermophilic bacteria called Thermus aquaticus. The order in which the free nucleotides are added is determined by the sequences of nucleotides in the original template DNA strand. The result of 1 cycle of PCR is 2 double-stranded sequences of target DNA, each containing 1 newly made strand and 1 original strand. This cycle is then repeated many times during the process of 2 hours, usually around 20 to 30 times, as most processes using PCR need large quantities of DNA. It only takes about 2 to 3 hours to get around 1 billion copies of DNA. To get started, we're going to need the following: the positive DNA control, our DNA samples, the nuclease-free water, the Taq polymerase, which is the DNA polymerase that is gonna join the free DNA nucleotides together. We're gonna need the forward and the reverse primers, and also make sure to use the proper size pipettes. So when turning in our thermocycler, we're gonna go ahead and use the protocol library. And in this case, you're gonna see all the protocols related to different type of users, persons that work in your lab. So in this case, let's just choose. I'm gonna be choosing the ESBL Monstein protocol, which is a protocol, right, to detect the presence of beta-lactamase resistant genes in some gram-negative bacteria. So in that case, we're gonna go there. We're gonna choose ESBL Monstein. So we're just gonna press enter. We're gonna press run protocol. In this case, you just have to choose if you're gonna use the right one or the left one. So that's pretty much the question that is asking you. So in this case, we can choose just the left one. Hot start, we're gonna press no. Hot start temperature, we're gonna leave it at 95. We're just gonna leave it as is, as the basic standard procedure that is part of that protocol. And in this case, you're just gonna put the amount of the microliters that you were able to put in your PCR sample. Since we use 25 microliters, we're gonna go down and put 25. So in that case, it's ready, and you just need to press begin run. You're gonna see the degrees that are getting started. They're getting to, they get higher to get to the ideal initial denaturalization process of the DNA. And let's just remember that the DNA needs to break apart, right? Break loose. So the strands, the DNA strands can be open and begin the replication process. So in that case, you just need to wait for it to begin to reach out to ideal temperature. So in this image, we can see that 3 different steps of the PCR process. The step 1, the denaturation, the step 2, the annealing, and the step 3, the extension, as the image that we were explaining earlier. In this case, you can see the cycle is beginning with 1 out of 3. So this is going to be continuously repeated until it reaches like a total of 30 cycles, and when it's completed after a process of 2 hours. Once our samples are ready, we can proceed to do the gel electrophoresis.
Let's talk about running your gel in the electrophoresis. There are 2 important things that you must consider. Number 1 is voltage, and number 2 is time. With the voltage, you gotta remember that the higher the voltage, the faster your bands are gonna run on your gel. Now, if you just want to determine like the presence of the gelling of DNA present on your gel, that's like a good idea. If you just want to be able to see if you got the presence of a good DNA sample, that's good. But for example, if you want to be able to have, if you have like different bands, or if you're running like a multiplex in the same line, you're running like different gene bands, then I recommend to you to use like a lower voltage. Even though it's gonna require like a longer time for it to run, you're gonna have like a very clear and very a good bands that are visible on your gel, especially if you want to be able to use them, for example, for your like a poster presentation or even like a talk, and you need like a very good image of the bands that you got. I recommend to use like voltage of around like 65, 70 ideally. And then, for example, for the time, the longer the time, the slower the voltage, then the nicer bands that you are gonna be able to get on your gel. So remember that lower voltage but longer time. So this is when we're talking about the time, right? For example, P2 usually ideally represents which one would you be using, the number 1 or the number 2. For example, the time, I like to run like if I want like a really nice clear image, I usually go for like 90 minutes. So I do take my time. For the voltage, and we can usually go for less. Ideally, I will recommend maybe 65 or 70 of the voltage. Yes, there we go.
So at a 70 of approximately 90 minutes, usually it's gonna give it like a good time. So for the materials, we're gonna need the pipette and the pipette tips, the samples with dye, the ethidium bromide, the gel box, a flask, and a cylinder. We're also gonna need the molecular agarose for the gel. We are also going to need a buffer. In the case of electrophoresis that separates on the basis of charge, ions in the buffer transmit the charge necessary for separation. The buffer, by providing a reservoir of weak acid and base, also keeps the pH within the narrow range. This is important because the structure and charge of a protein or nucleic acid will change if subjected to significant pH changes, then preventing proper separation. Ethidium bromide is particularly useful to stain DNA in gels, but it is very important for you to remember to wear gloves because ethidium bromide is considered a very carcinogenic substance. Okay, so we're gonna remove this from here. Once it's ready, we're just gonna put it in our buffer. Okay, so we're gonna put our gel in our electrophoresis case, ready with our buffer 1TX. Remember that, and it's also good to remember that the voltage, that the way it's gonna run is gonna be from negative to positive. So you want to be able to have your positive facing you. Once you know that it's going to be running, you're going to detect some bubbles coming up from the negative side of the case. Now, choosing the right molecular ruler as part of your gel is also like a very important step. It can go from 20 base pairs to 1K base pairs, but it also determines it depends pretty much of the size of the gene that you're looking for on your gel. So mostly, for example, if I'm looking for one of the KPC genes, right, of resistance, it's gonna be around 200 BPs. So for me, ideally, I love using the 100 BPs because it's gonna tell me on my gel the size specifically that I'm looking into my ruler. It's very important for you to know already what is the size of the gene on your control and also on your DNA samples, because that way you're gonna be able to determine if that corresponds to the correct gene that you're looking for and that you're getting in your gel sample. So now we are ready to put our samples and our molecular ruler into our gel. I usually recommend first you're gonna put, of course, your molecular ruler as your line number 1, because that's what you're gonna compare the next DNA samples. I usually go with the molecular sample, then I put my controls, positive and negatives, and then from there I put my DNA samples. So it will give me an idea of what am I comparing my DNA samples to, and I have my positive control, and they should be able to be on the same line on that when I'm analyzing my gel. So I also recommend, for example, that you have like a steady hand. Sometimes it can be like very tricky, right? Some beginners, some students might be able to go through the gel. So having like a steady hand and then using one steady and then one like to insert the samples also gives you some control over your gel once you're putting them into your gel. So first we're gonna do our molecular ruler. I usually put around 4 microliters
of the ruler into my gel. I think that's a good quality that I've seen. So we're gonna go ahead, like I was telling you, hold, put one for control, like to give you some resistance, and then with the other one you're gonna be going over and pushing into your gel, right? So as you see, you're gonna be putting your number 1 on your first space. You're gonna press and you're just gonna release slowly your sample, and then you're just gonna take out, and you're gonna see the number 1 on the 1 space. Our molecular ruler is right there. For the number 2, I'm gonna be putting my control, my positive control, and you see you're gonna put it right there on the next space, and you're just gonna release slowly your sample, and you're just gonna let go again. Pretty much the molecular ruler I'll always have like a blue color, and then the PCR ones, because of the tag, it has like a slight green color that allows you to visually follow the rules when the gel is running. We go ahead and insert the cables connected to the electrophoresis, and then we're just gonna go ahead and run. It's ready to go. Press this button right here. So you're gonna start noticing the voltage starts going to the number that you set it up in the beginning with. And that's it. We just have the gel ready, and we just have to wait for 90 minutes for it to run completely. So once the gel is ready, we're just gonna pull it out just like that. Just try to get rid of that extra buffer that we have there, and that means that your gel is ready for analysis on the fluorescence lab.
And here we have a final image of the gel. On the first line, you're gonna see the lines of the 100 pp's of the molecular ruler. Then we're gonna see 2 positive controls, and then we're gonna see a line of that belong to the same size of DNA, and that's our DNA sample. Sometimes you will see like a difference on the color of the DNA, but that depends pretty much on the quality of the DNA extraction, and also on the concentration of the DNA that was able to be recovered after the DNA extraction.
That's it for today. Thank you so much for watching this video. I really hope that you were able to learn and understand how to do a PCR and how to do a gel, and you follow these steps so you can do one for yourselves in your own lab. Please let us know what video, what lab technique you would like to learn, so we are able to do a video for you and you can apply all these lab techniques in your own lab. Thank you so much.